These notes are on forensic serology, which is the study of blood. Specifically, this is going to be about blood composition. So, what blood is made of. So, blood is a mixture of cells, enzymes, proteins, and inorganic substances. So more specifically, it's got kind of two main parts. So plasma is the fluid portion. This is just over half of your blood. This is mostly water with some proteins, sugars, salts, etc. in there. The solids then, so just a little less than half, these are where you're going to find your cells, like your red blood cells. So these are also called erythrocytes. The white blood cells, which are your leukocytes, and then platelets, which help your blood clot. So if you get a cut, you won't just keep bleeding. Platelets will make sure you stop bleeding at some point. All right, so Karl Landsteiner was an Austrian biologist and physician. He studied and found that human blood has four main blood groups in 1900. So he was able to see or find out that there were specific markers on the red blood cells, the erythrocytes. So the blood types were A, B, AB together, and O. Now, in addition to that, each of these can be either positive or negative. That's showing that they have the Rh antigen. So if they have it, it's positive. If it doesn't, it's negative. And about 85% of Americans are positive. So your red blood cells are where you're going to find these. Their primary function is to carry oxygen to your organs, different parts of your body from your lungs, and then they'll come back and get more oxygen. They contain hemoglobin, it's the protein that oxygen binds to. So the surface is where you're going to find these antigens. That's what determines what blood type. The antigens stimulate the body to produce antibodies. And the antibodies destroy or inactivate antigens. So, for example, they're going to attack blood that is not the same type as yours. So if I have type B blood, I have antibodies for A, so that my body, if I get type A blood, my body is going to attack it because I can only have B or O, which we'll get to in a second. So the four blood types, A, B, and O, A and B are co-dominant, so meaning they can both exist together. O means you don't have A or B, so it's recessive. So a person with the genotype with the two capital I's and the A capital A's there, that would be, or AI, so capital A and lowercase I, these are both going to show type A blood. So the capital I is when you have a dominant gene and a lowercase I is the recessive gene. So if you have a dominant with another dominant, they will both be the same or coexist or co-dominant if it's A and B. If you have a dominant and a recessive, the recessive will not show up. That trait won't show up. All right, so you can see these are the four blood types. We're not talking about positive and negative quite yet. But we've got, if you have type A blood, you can see you've got these type A. Well, let me get a pen. You've got these A antigens right here. Those are the little circles on here. All right, so if you have those, it means you have anti-B. So if you have type A blood and you get a transfusion of B, this anti-B right here is going to attack the blood. And it's going to cause problems, which we'll get to. It's called this agglutination, okay, where your blood will clump up. All right, so if you're A, you can get type A blood or O blood, because if you look at O over here, O has no antigens, none. So it has anti-A and anti-B, but if you don't have, but it doesn't have any of those. All right, so a little bit more in depth, like here's a red blood cell, for example, we've got drawing. So an A antigen in all of these models are gonna be a little square, a B antigen is gonna be a triangle, and the RH is gonna be this funny little U shape. So the antibodies will attack it if it exists. So let's go. So here you can see RBC is for red blood cell. A is the squares. So these both have type A blood. Positive means we've got the RH factor right here. Negative means there is no RH factor. So B, same idea, right? B positive versus negative. They both have the B, right? Antigen, positive also has the RH factor. All right, AB, codominant. These both exist. So here's your A and B. A and B. Positive has the Rh factor. O has neither A nor B. O positive has the Rh factor. O negative does not. So O negative has 0 antigens. All right, so agglutination is the interaction when you've got the antigen with an antibody and you have visible clumping. All right, so blood agglutination comes from the presence of foreign antigens. So antibodies in your body will fight foreign things. Okay, so this leads then to your genotypes and your phenotypes. So genotypes are the genes that you have. You get one from each parent. So genotypes, you're always going to have 2. Phenotype is what trait is shown. So your actual blood type. So you can see in the green box here, I've got genotypes of AA, so 2 dominant, or AO, dominant and recessive. You're going to get a phenotype of A. Same with B. You can have 2B or 2B dominant genes, or a B and an O, a dominant and a recessive, and you're going to get type B blood. AB blood you can only get from AB, so codominance. And then O blood you can only get from 2 O's, so 2 recessive. All right. So you can see it kind of in the other tables, same thing, just another way of writing it with capital I's for dominant genes and lowercase I for recessive genes. So notice A and B, there's 2 possible genotypes. So they call these homogeneous and homozygous and heterozygous. Homo meaning the same. So homozygous would be like this one versus heterozygous, which would be like this one, where they're different. Okay. A and B blood, though, you can only get from one genotype. All right, so frequency. Most common is O positive. Next most common is A positive. Then we get into quite a bit more rare blood with our O negative is 9% of the population. B positive is 9%. Then we go to A negative. The most rare is AB negative. All right, so how does it work when you need blood or when you can give blood? So if we look kind of in this middle row right here, all right, we can see the blood types. If you have O negative blood, you can only receive blood from someone else who's O negative. So you can see the arrow right here. Okay, you can only get O negative blood. However, O negative blood can be given to all other blood types. So O negative is the universal donor. So if anyone needs a transfusion or blood at a hospital and they don't know what their blood type is, they will give them O negative because that is safe, because O negative has no antigens, so it has no antibodies to attack anything. All right. So you can kind of look through the rest of these. AB positive on the other side. AB positive can get any blood, but it can only donate to AB positive. Okay, so what I want you to notice is O can receive blood from any O. Notice like the A's and the B's. A can receive from A, but it can also receive from O. Same with B. B can receive from A and O. AB can get AB blood, A blood, B blood, and O blood. But then look at the positive versus negative. If you're positive, like let's look at A positive, for example, you can see they can get A positive or A negative because negative doesn't have the antibodies to attack it. Same with O positive or O negative. Same with B positive. However, the B negative or A negative cannot get a positive. All right, so determining blood type using Punnett squares. So this is looking at the genes from both parents. You can predict the relativity or the percentage of each blood type, like the possibility of it. So for example, if parent 1 has heterozygous A blood, so that would be this right here. Heterozygous, meaning we've got a dominant and a recessive. And parent 2 has heterozygous type B blood. That would be these right here. What are the possible genotypes? So first step is to identify them and write them in, like I just circled the green and the red. Then you're going to, and then place them in there. Then you're going to cross them. By crossing, we mean if I take this and this, the A and the B, right, one from each, here's what they would make, right? So this child could possibly have AB blood, okay? Then they could also have a child that has, right here, they could have B with the recessive. So where they combine is this. So that would be type B blood. You could get the recessive with A and have type A blood, or even 2 recessive and have type O blood. So this combination of parents could have a child with AB blood. They could have a child with, from this one. They could have a child with just B blood from this one. They could have a child with, oops, that's supposed to be a B, with just A blood, like this one. And they could also have a child with type O blood, right, from the 2 recessive. So it's a 25% chance for each blood type from this cross. And we will do more examples and practice with this in class.